When a person unexpectedly comes face to face with a grizzly bear, his or her body quickly shunts blood away from the skin and digestive system and toward the muscles. The heart also beats faster and the liver releases glucose molecules that provide emergency fuel for what is called the fight or flight response.
In the fight or flight response, the adrenal glands release the hormone epinephrine, which serves as a signal within the body. Certain cells, including liver cells, can detect the signal, after which they process the signal and respond to it. The entire sequence from signal reception to cellular response is referred to as a signal transduction pathway. The following animation depicts a signal transduction pathway in a liver cell.
Bro, no, the first step in epinephrine signal occurs when the hormone binds to epinephrine receptor on the cell surface. The hormone triggers the receptor to change shape to where a receptor to its active form.
Hey, yo!
It's not in its active form.
The activated receptor triggers a cascade of events within the cell, beginning with the activation of a G protein. The G protein binds to the activated receptor, releases GDP, and takes up a molecule of GTP.
After taking up GCP, the G protein is released from the receptor and splits into two parts. One of the parts is activated and continues the signaling cascade. Soon the hormones also leaves the receptor and the receptor reverts to its inactive form.
The G protein activates an enzyme called adenylylsilase. When activated, adenylylsilase converts a large number of AT molecules into signaling molecules called cyclic AMP, CAM, because CAM carries the message of the first messenger into the cell. CAMP is referred to as a second messenger.
In response to an internal timer, the G protein soon inactivates itself by cleaving GTP and the subunits reassociate. With the G protein no longer attached, the adenylyl cyclase turns off and can no longer convert ATP into CAMP.
But then I just had to like put that up.
The CAMP molecule produced by adenylyl cyclase continue the signaling cascade by binding to a type of enzyme called protein kinase A. This binding triggers protein kinase A to separate into subunits, two of which are catalytically active.
Go.
He played?
Oh, I think they mean in general.
No. Okay.
The activated protein kinase A subunits perform chemical reactions in which they add phosphate groups to another type of enzyme called phosphorylase kinase. The addition of the phosphate groups activates phosphorylase kinase.
I don't care. To which are catalytically
I was not recording in time. Can you say?
I am a lost boy from Neverland, usually hanging out with Peter Pan. And when we're bored, we play in the woods, always on the run from Captain Hook.
Phosphorylase kinase and phosphorylates another enzyme in the cascade called glycogen phosphorylase. When phosphorylated, this enzyme also becomes activated.
In its activated site, glycogen phosphorylase produces the cellular response in epinephrine. Glycogen phosphorylase breaks down glycogen into its component glucose molecules during the process. The enzyme adds a phosphate group to each of the glucose subunits.
It's just a paper punches.
That is actually good. That is so good. That was actually so good.
I can't do it again, man. If I do it again, it's gonna mess with my record, bro. Are you showing me? Another enzyme removes the phosphate groups from the glucose molecules.
Is it phosphate? You could just have them go like this type of thing. Yeah, and then mush it together.
Without a phosphate group, glucose molecules can be transported across the plasma membrane of the cell. Once outside the cell, glucose enters the bloodstream and is taken by other cells and used as a fueling key component of epinephrine and induced flight response.
Go.
Signal transduction pathways allow cells to respond to environmental signals. In the majority of signal transduction pathways, the signal is amplified such that most steps produce a larger number of activated components than previous steps. Signal amplification, for example, results in a liver cell releasing many glucose molecules after detecting a single molecule of
epinephrine.
Signal amplification can occur at many points. For example, as long as epinephrine remains bound to a receptor, the receptor can activate a succession of G proteins. In addition, each adenylyl cyclase enzyme can convert numerous ATPs into cyclic AMP molecules. Other activated molecules in the pathway can also continually catalyze reactions. The G protein, in contrast, activates just a single adenylyl cyclase enzyme and must remain attached to it in order for adenylyl cyclase to remain activated.
Termination of the cellular response is as important as its initiation. In order for a cell to respond only when a signal is present, the many players in the pathway have to be regulated so that they are activated.